Hello, everybody. Good morning and good afternoon, and welcome to today's webinar titled Advancing Early Detection of Colorectal Cancer with Novel Blood-Based Protein Biomarkers. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Olink Proteomics. To learn more, visit olink.com. As a reminder, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will go ahead and answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome Valentina Menegan, Global Marketing Manager at Olink Proteomics. Vale, over to you.
Thank you so much, Sydney, and hello everyone, and welcome to the last Olink Multiomics Coffee Break of the year, organized in collaboration with Protavio, an Olink certified service provider and also authorized distributor for Greece, Cyprus, and Malta. So as Sydney already introduced me, I'm Vale Meneghin, Global Marketing Manager here at Olink, and I'm pleased to give you a quick overview on Olink Proteomics for those of you who are new to our company or technology. Okay, so Olink is a Swedish company by origin that in a few years has grown rapidly from a small Sweden-based company offering a safe for a few hundred proteins to a global leader with a broad portfolio of flexible protein biomarker solutions and a library of high-quality, thoroughly validated assays that cover more than 5,000 proteins. Our customers continue to drive significant advancements in the field with over 3,200 publications to date, covering a broad range of application and disease areas. And as you may have heard, we are really proud that the UK Biobank Pharma Proteomics project chose the Olink Explore HT to measure over 5,400 proteins in 600,000 samples from the UKB participants, so the full database. And this represents the largest ongoing proteomic study that aims to create the most comprehensive map of protein levels in the human body, significantly advancing our understanding of disease. And of course, this is indeed in line with the ambitious mission of Olink of revealing the truth of human disease protein by protein. But let's say that we are not alone in this mission. So we are doing this together with our customers, the research community, and with a strong network of certified service providers. And we are really proud to present to you the Olink CSP network that is equipped to honor our commitment of delivering excellent data to our global research community. So the Olink certified service providers are trained by Olink scientists and undergo annual recertification through concordant testing. And this is the reason why they can help you to accelerate your research, avoiding any roadblocks and facilitating the integration of the cutting-edge proteomic solutions into your studies. And we are really proud that Protavio is part of this prestigious network, and this ensures that you can confidently outsource your projects to them. Now, without further ado, let's move on to the agenda for today. So I'm pleased to welcome Christos Fotis, CIO at Protavio, who will present Protavio's services and opportunities, and Professor Leonidas Alexopoulos from the National University of Athens, who will share this amazing case study that highlights the absolute key role in cancer early detection of protein biomarkers to advance oncology research. So after Dr. Alexopoulos' presentation, we will have a Q&A session moderated by our very own Pasquale Cutolo, Olink Senior Field Application Scientist. So I invite you to add all the questions you may have throughout the webinar in the Q&A box so that we can take them at the end of the session. And now, without further ado, I will hand over to Christos.
Thank you, Vale, for the nice introduction, and it's nice to see you all here. At Protavio, Our vision is to improve people's health through innovation in proteomics, and Protavio specializes in multiplex protein biomarker assays to offer services for protein biomarker discovery, bioinformatics, as well as orthogonal validation of biomarkers, and the development and manufacturing of multiplex protein diagnostics for early disease detection. Protavio is a Greek company that started in 2012 at the height of the Greek economic crisis with only 5,000 in capital, without any access to VC, so we had to go directly to the market to grow. We established our first lab facilities in 2013 at the Democritos Science Park, and following that, our first GMP manufacturing facility for multiplex assays in 2018. And we received our first ISO certification for production in 2020. We're very lucky to start in 2022 to deliver the first Olink discovery facility in Greece for Olink Hyplex to discover new protein biomarkers for early disease detection. Over the year, Protavio has extensive expertise in multiplex protein biomarkers, and we have completed over 200 projects in this space. In terms of Olink and our collaboration with Olink, we are certified providers for both Hyplex technologies, both Olink Explore HD and Olink Reveal. And as Vale said, we are now also distributors of Olink reagents and Olink instruments in Greece, Cyprus, and Malta. We have experience from all these biomarker projects that we have completed in the past, and we can support our partners and customers with the clinical and experimental design of their projects, as well as the analysis, in order to maximize the translation potential of the biomarkers. And on that, we can also assist with SOPs and best practices regarding sample collection and processes to minimize pre-analytical variations. We also have a very strong bioinformatics group with the main goal to prioritize biomarkers, protein biomarkers with optimal performance, both as individual markers and as a combination. And on that, first we assess the biomarker performance in a single format, then we prioritize mechanism-based biomarkers, and then we try to find the optimal biomarker combination using machine learning models and logic-based models. And on that space, we have more than 100 publications for multiplex biomarker combinations. Now, we also place a strong focus on the quality of our services and products, and we have a quality management system at the company that is certified for ISO 9001 for multiplex protein analysis and ISO 13485 for the development and manufacturing of multiplex protein diagnostics. Using this quality system and our experience, we have developed and delivered a 10-step pipeline to translate novel protein biomarkers that we discover or our partners discover into multiplex IVDs that can be used in the clinic. This pipeline includes the orthogonal validation of the biomarkers, the development of multiplex assays under the in vitro diagnostic regulations, and finally the production of the kits for IVD or LDT use. And currently, Protavio produces more than 1 million tests in multiplex format annually. We also participate and are involved in R&D within the Horizon Europe and the Mission Cancer Initiative, where we contribute to 2 key initiatives. First, the Dioptra project, for which you will hear your presentation that includes some of the results of this project for colorectal cancer screening, and the SILK project for pancreatic cancer early detection, where our goal is to deliver novel protein markers for prevention and early detection of cancer. Our main R&D facilities are located in Athens, Greece, and now we're expanding a new space specifically for the GMP production of multiplex assays. And Protavio's offices are located in 3 science parks in Athens, Greece, Boston, US, and Cambridge, UK. And with that, if you have any interest in working with Protavio for your biomarker projects, please contact us directly, or you can scan the QR code to submit your request. Thank you for your attention, and I'll pass it now to Professor Leonidas Alexopoulos for his presentation. Thank you very much.
Thank you very much for the introduction. It is my pleasure to introduce you about the recent work that we've been doing on using Olink for advancing early detection of colorectal cancer. So before discussing about the colorectal cancer, I would like to mention the two hats that I'm working on. I mean, I'm both a professor at the National Technical University of Athens, and I'm doing research in my lab on systems biology here in Greece, but also for 10 years in the US. And but I have seen the last 15 years via Protavio how we can use research findings to develop multiplex diagnostics, especially for today's talk about colon cancer screening using the Dioptra EU project. What we have seen in these 200 biomarker applications that we have done and what Christos mentioned is that despite the fact that 60% of them, they have strong biomarker findings, very few they translate into real IVD products and they follow a path to commercialization. So there is a real gap between translating research to a diagnostic product. And if I wear the professor's hat, a professor is interested for a high impact journal papers. He receives funding and the output is papers or intellectual property. And it's a cycle for getting papers out, receiving more funding, citations, reputation, and so on. But from the industrial point of view, what you really need is a clinical need. And from the clinical need, you need to have the regulatory requirements, the ISO 13485 manufacturing, the development, the real-world validation in order to commercialize it. So at the end of the day, you have an intended use, and you're focusing on approving the intended use through the regulatory pathway, which is the intended use that is your focus on the IVD. In many cases, is not the same as your research focus that was a high-impact publication. So you can see like some gaps, and this is also why a lot of biomarker projects do not lead to a product. Sometimes it's a business reason, there is no clinical need, so you have a contrast between normal and disease, but there is other methods that the doctor can take a decision from imaging or from something else, so there is not a real clinical need for this blood-based biomarker. There are also other regulatory and funding bottlenecks, stakeholder interest, ISO manufacturing, and so on. But from the scientific reasons, the initial biomarker findings that you do, like, for example, a normal versus disease, do not support the intended use. You start thinking about the intended use only when you start dealing with IVD manufacturing. And something that we're going to focus today is pre-analytical variability. At the end of the day, the requirements for high-impact publications are different, but they can be aligned with the regulatory requirements, so the time and the effort that you're spending on your research can be easily translated into a diagnostic product. So as Christos said, the 10-step process from discovery to clinical use, first of all, is discovery, and we are heavily using Olink on that. At the end of the day of the discovery, you need to have an intended use to do your analysis, find your biomarkers, but at the end of the day, you have a list of potential biomarkers. Then is the design and development phase, where you're developing the assays in the platform that you are choosing, and it fits to the intended use. You have to find a good algorithm to select platforms, but on the end of the day, you have a final plex, a 5-plex, 10-plex, and an algorithm for production. Then when everything is said is what you do design freeze. And when you do design freeze, you're moving to the manufacturing. So at the end of the manufacturing, you're going to make the kit and the software, not the algorithm, the software that is ISO 13485 compliant for commercialization. And the last phase, of course, is the commercialization, the diagnostic use as a RUO, research use product, as an LDT or as an IVD. So focusing now on the colorectal cancer. Colorectal cancer is a type of cancer that affects the colon or the rectum. It is one of the most common types of cancer worldwide. And the development of CRC, colorectal cancer, usually starts with a small hyperplasia, it becomes dysplasia, adenoma, advanced adenoma, and then it goes to cancer 1, 2, 3, and 4. And there are a lot of well-established molecular mechanisms. It can be chromosomal instability, genetic mutations, hypermethylations, and so on. For the blood-based biomarkers, there is a hypothesis. So, and specifically for the colon. So a small lesion that we have, it can induce a big signal. What is this big signal? You have a small piece of tissue, let's say a small piece of tissue is 1 cubic centimeter, approximately 1 ml, if it was water or 80% water anyway, and interacts with adjacent tissue to release some detectable molecules in the 5 average liters of blood. And this biomarker signal can be from live cells or reactive. They're reacting with tumors and adjacent cells, and they have an immune reaction. And then at the end of the day, you can detect proteins in the bloodstream that is related to communication, hallmark of cancer, host immune response. It can be mucin-related glycoproteins, like very well-known cancer antigens. It can be antibodies, autoantibodies, from abnormal proteins from the tumor cells or from cells that they have died, and our immune system recognizes them as a foreign body and develops autoantibodies, like, for example, P53. It can be extracellular vehicles, exosomes and microvessels, or these signals can come later when the tumor cells have started dying and they're releasing things into the bloodstream. That can be intracellular proteins or from normal or mutated proteins, cell-free DNA, immune-reactive proteins or autoantibodies from dead cells, and so on. And depending on the platform and what biomarker you have to choose, for proteins, Olink is an amazing platform. For NGS, you can use also molecular, you can combine, depending on what platform you end up using at the end of the day. And there is also an AI now with the advancements of the AI. You can use also AI algorithms to identify new biomarkers. So the key parameters to consider when you are doing when we have done the colon cancer discovery, it's not a single fold change between CRC and healthy or a p-value. Proteins compared to cell-free DNA or mutated DNA can be increasingly not specific biomarkers because their mechanism is shared among other biological processes, and there is a lot of confounding factors. There is a lot of pre-analytical variability that we're going to discuss later, and you have to do appropriate study design. So it's not CRC versus healthy, but it's CRC versus, if it's for screening, it's for the average risk person that has negative colonoscopy confirmed. And this is very important, and to the extent that you can do it on the screening, you have to use also the prevalence of the disease. So you expect that this assay will be used in public, it will be used for people that only 0.6% of them are going to be positive, but the same people are going to have many other diseases that might have a stronger prevalence than the colon cancer. So thinking about that, we were thinking of starting in Greece and having this process in Greece. So you have the population like 3 million people. Right now, a lot of them are asymptomatic. They're receiving an SMS from the government, and they go and they do the fit test. It's a self-fit test. If this test is going to be negative, they're going to come next year. If it's going to be positive, they're going to go for colonoscopy. Depending on what country you are, you can pay from 100 to 3,000. And then from the colonoscopy, there's going to be a staging, and hopefully you're going to be on polyps or early stage, so you have a very high survival rate, and not on late stage that you have a very low survival rate, 5 years. There are people that they're not going to do the asymptomatic process, and they're going to have some symptoms, and usually when you have symptoms, and you act upon a symptom, and you don't act upon an SMS or something that you go and you do it with asymptomatically, usually you detect colon cancer in late stages that has a lower survival rates. And what we would like to do in this case is to replace the FIT test with a test that many people, hopefully a very simple blood test that there is high compliance that many people can take. These are many other available early detection tests. You can split on stool-based test or blood-based test, and they can measure either proteins or DNA-related molecular assays. So for the DIOPERA project, we would like to revolutionize the colorectal cancer screening. We have 28 partners in Europe, 15 countries, 9 clinical sites. And what we would like to do is to match plasma serum samples and tissue and be able to find biomarkers that are related to the mechanism. On this front, there is 8 hospitals. In total, we're collecting almost a little bit more than 3,000 samples from hospitals in Greece, Cyprus, Slovenia, Croatia, Belgium, Denmark, Austria, and Spain. What is important to mention is that we were thinking of the scenario that at the end of the day, when this assay is up and running, there are going to be many different sites in Greece, but also in Europe, that you're going to take the samples and you're going to collect it, and you're going to ship them to central labs or to local labs. That depends on the platform that you're going to use later on. But in this sample analysis workflow, you are very careful that the first step is to go to the phlebotomy room. You take the venous blood draw, either with a syringe or butterfly. The second step is to put that blood in a serum or in a plasma with gel separators or activators, and then to mix the tubes, to leave it on a rack, either for clotting if it's serum, or to leave it in a rack at the phlebotomy room, because before that person takes this rack and brings it into the lab. And then you go into the lab, you centrifuge your sample, you're going to aliquot it and create triage routing, so maybe at the same time you need to measure cholesterol or some other things. You store it either at 4 degrees if you're going to measure it immediately, or you ship it in -20 if you would like to ship it somewhere else. And then you can either ship it on the same city with a van that moves around at 4 degrees, or you can send it with dry ice or at 4 degrees, depending on the platforms that you're going to choose and the workflow that you're setting up, to different, even to different countries. So all these steps create some pre-analytical variability, PAV, we call it PAV, from pre-analytical variability. And how we have done the discovery phase, initially we're using Olink to explore with a pre-analytical focus, and that's what we're going to discuss now. But identifying the pre-analytical variability, it makes it easier to design a study to focus on the disease and see the real CRC effect. So the pre-analytical variables from the process that we discussed before is either you're going to use plasma or serum, your tube selection, the rack storage, how much time you're going to leave it in the rack, the centrifuge speed, shipment requirements, clinical sites, how many clinical sites and what SOPs the different clinical sites have for the blood collection, and some other also pre-analytical variables. So the first phase is to see like the CRC versus the PAV effect. So the cancer effect versus the pre-analytical effect. And it's a study with almost 300 individuals. 60 of them approximately have CRC. 100 of them is advanced adenoma. There is also people that they have a non-advanced adenoma and they are negative. And the variables that we're taking on top of the CRC versus negative or non-advanced adenoma are plasma versus serum, time to centrifuge, and the centrifugation specifications. Initially, when you just don't look into the pre-analytical variables and you just see a simple like a volcano plot, you see like a nice volcano plot, and in this volcano plot you can identify from 25 up to 50 different biomarkers that they are good for CRC detection. And when you are combining them, you can see like individual the performance of each protein. But with this rock analysis, you can combine it bioinformatically in a single value, you can have better performance. So by doing that, initially we were able to identify a sensitivity like of 96 and specificity of 93% for CRC, and of course lower for advanced adenoma. It's not easy to find advanced adenoma. 37% sensitivity and 93% specificity. So protein biomarker signatures in blood can be used for detecting CRC, but how many of these biomarkers are real biomarkers and it's not like a PAV effect? So we start exploring more using the Olink reveal, And on these plots that you see here, we compare the rack storage time, how much you leave the cube in the rack before goes and before simplification. And something really important that we saw is that the serum samples, they have a lot of variability compared to the plasma samples. So the serum samples seems to be prone to cytokine activation, probably because of PBMCs that they're releasing a lot of stuff during clotting or during rack storage time. And if you compare that, how erupted is this volcano plot compared to how erupted is the volcano from the CRC effect, you can see that the PAV effect is stronger than the CRC effect on the serum sample. And in real world settings, when you use that in very different labs, rack storage time is very hard to control. You can include biomarkers that you can exclude, like things that probably DPMCs are releasing. You can have a plex to identify the rack storage time, but we can discuss that if there are any questions. So the other effect that we wanted to see is that when it moves from the rack and goes to the centrifuge, if the centrifuge specifications are good, so and are affecting the proteins, the released proteins. And here we found that the plasma versus the serum, that the plasma samples are affected by centrifugation, not so much as the serum, but when we compare 500Z versus 1700Z, we saw that some plasma samples, they had some released proteins that they were due to the centrifugation speed. And this effect, this additional centrifugation PAV effect, is similar to the CRC effect. Similar means that not the same proteins are affected. So having like a PAV effect, that does not mean that the CRC effect is not good. You would like to select proteins on the CRC that they are not present on the PAV. And you can find a lot of proteins that they are different between the groups. So these are very good news. We were surprised by this finding, and we did another experiment that we had no centrifugation, just to see if there is no centrifugation, extreme case. I mean, this is not happening in the real life, but it was extreme case to see like the real, how bad the PAV effect can be. And really, like the PAV effect can be really bad, because here you can see, for example, the PAV effect compared to the CRC effect. So when you have no centrifugation of plasma, it is very significant, and that can be due to the PBMC activation. I mean, you can lyse also PBMCs. It's not a thing that really happens, but we have to be careful, and it shows that how PAV effect can be a strong effect. So the conclusions of the pre-analytical variability is that the pre-analytical variability can have stronger effects than the proteome compared to the CRC effect, and that makes sense scientifically. I mean, you have a small piece of tissue, 1 centimeter. Okay, it can release some protein, but the PBMC sitting like in a tube can also release a lot of different proteins. Plasma samples are more stable, they have lower PAV effect, and that was a very important key point that later on, when we did the discovery, we used the plasma samples. And potential biomarkers for CRC or advanced adenoma that we found from the CRC effect needs to be filtered out due to the PAV effect. But if it's a good biomarker and you don't want to miss it, then you have to compensate it from the PAV effect. And other PAV effects also might be present. And at the end of the day, what you would like to do is when you're running your clinical trial, you have to collect samples from many different clinical sites, many different phlebotomies, so you can minimize this PAV effect as much as possible. So by saying that, we're moving to the second phase, which was the disease-focused panel. And for the disease-focused panel, we need to use as many proteins as possible, and that's why we're using the Olink Explore. So for the Olink Explore, we have now almost 200 participants, CRC, advanced adenoma, non-advanced adenoma, and negatives. And we used also some healthy donors just to increase the healthy donor variability. And the key points here is that we use plasma based on the PAV analysis and many different clinical sites, which is the real world scenario. And also we did audits to address SOP discrepancies between the clinical sites. So using that, using the Olink Explore, we found a very good performance. Depending on how many proteins we're going to use, we found 120 biomarkers. Later on, depending what platform we're going to use, we're going to miss a lot of them due to the analytical sensitivity issues and platform specifications. But using 27 of those biomarkers, we found the sensitivity that you see here, 100% and 93% sensitivity. And for advanced adenoma, we were able to find 47% sensitivity and 93% specificity. And the last but not least is the additional biomarkers. So AI is giving you an amazing tool to search for the biomarker you found from Olink, to manually AI curate it and see like if they make sense mechanistically. But also you can search for biomarkers that they are on the literature and they are not on the Olink panels. So they're not on the 5,000 panels that so we searched the AI-based the literature. We found 54 additional mechanistic biomarkers that in the literature they claim that they can be good for the CRC diagnosis. 28 of them are hallmarks of cancer. 26, we focused a lot on the autoantibodies because autoantibodies was not something that was present on the Olink panel, and we wanted to enhance the sensitivity and the specificity of the test. Autoantibodies are serological assays, so if we're going to end up using bit-based assays, we can put them on the same process, and we can have panels that they have exactly the same sample prep, and some of them are proteins, some of them are autoantibodies. So we found a lot of, from the 54 different biomarkers, we did ELISA, single ELISA, to validate them whether they're good or not. We found some protein markers and some traditional markers that they are very, like SEA, that are very well known for cancer. And for the autoantibodies, this is a little bit different story. You don't expect a lot of patients, like all the cancer patients, you don't expect a lot of cancer patients to have autoantibodies. You expect like 20% of them that are going to have autoantibodies. So you have to have a different algorithm. The autoantibody, if it's on, it can be like 6 deviations or more. It can be very, very, very significant upregulation. And you have to use a carefully selected decision algorithm. So if it's an outlier, then you can claim that this can be like a colon cancer case. So the next step is once we identify what's going to be the final hits, we go to the phase 2 that we do orthogonal validation, development of other kinds of technologies, maybe bit-based assays. The phase 3 is to have the algorithm that we identified to make a diagnostic software, so to follow the ISO 13485. And then also the panel that we developed, to make it a diagnostic kit under IVD and ISO 13485, IVDR and ISO 13485 guidelines. And then the last phase, of course, is to test our Envision solution for the CRC screening with another European project, hopefully that we move forward, that we can. You don't need only 3,000 individuals, you need 10,000 individuals because your prevalence is like 0.6, 0.5%. So in order to have 50 cases, you need to screen like 9,950 people. And this needs a different clinical trial. So in conclusion, the protein biomarker panel shows strong performance in detecting CRC and advanced adenoma. Pre-analytical variability, we need to take it seriously. It can affect many potential biomarkers, and that can be either excluded because, or you can compensate it algorithmically. The biomarker discovery relies heavily on the selected samples. You have to be very, very careful on how you design your clinical study, and the clinical study should account for real-world scenarios and pre-analytical variabilities. And as I take home for the Olink panels, we can have quick results, like in a few days, from 1 day to 3 days, depending if it's the Explore HT panel or the 1,000 or the 5,000 panels. That can really help us a lot to accelerate the biomarker discovery. So if you would like to see something quick, like a PAV effect. You can run Olink reveal 1000K proteins, which is cost effective. If you really would like to identify like a novel biomarkers, then the 6000 panel is suitable for discovering mechanisms. And on top of that, with AI, you can validate the mechanisms of those. You can have a clear idea of the mechanisms of those new biomarkers. Without and closing, I would like to acknowledge all these people that they are present for in the Dioptra work that we presented today, and I'll be happy to take any questions.
Perfect. Thank you so much, Leo, for such an insightful presentation. Really interesting and highlighting the power really of protein biomarker in detecting early colorectal cancer. So now I remind the audience to place all your questions in the Q&A box, is the one with a question mark. So please add all of your questions there. And before starting with the Q&A, I would like to launch sample questions because for us it's important to understand if you have any question or doubt regarding or curiosity even on this project. But also we would like to learn a little bit more about you so that we can better plan the next webinars. So I start with the first question. So are you currently working on protein biomarker studies? Please reply with a yes or no. Don't be shy. Okay, now I launched the question, sorry. Okay. Next question. If you are not, are you planning to start in the next 6 months? Okay. Next question. So would you like to be contacted by an Olink rep to learn more about the Olink PA technology and how it can be integrated into your omics workflows? Okay, next one. Would you like to be contacted by Protavio to discuss starting your Olink project? And the last one, which application areas are most relevant to your current research? And we have different choices: biomarker discovery, clinical research, drug development, translational studies, or other. Okay, so thank you so much for giving us your inputs. And with that, I would launch the Q&A session. So I see that Pasquale is already on camera. So Pasquale, over to you.
Yes, I see already 3 questions in the box. So, so the first one is, can you please explain if I'm wrong? Biomarkers act as an agent which produce proteins, and the cancer cells like to digest that proteins, so we can identify the cancer cells early. Maybe Ganchupal, if you can explain better the question, because I'm not sure I understood.
I think I understood the question. I think what means that is what we discussed before, that you have a hypothesis that a single cancer cell grows and becomes like gets some survival advantage from the hallmarks of cancer, and then becomes a tissue, a volume of 1 centimeter cubic that you can detect. During this process, interacts with the environment. And sometimes it's getting killed, sometimes it's releasing protein, sometimes it's going to release VEGF to ask for angiogenesis. So the better you understand the biology of this cancer growth and the hallmarks of cancer, you better you are to develop mechanistic biomarkers. So just don't look only the contrast healthy versus normal, but try to think, hypothesize how this tumor relates to the microenvironment, and how that proteins that they are releasing to communicate or die eventually, because some cancer cells are conducting apoptosis, and from the apoptosis they are releasing a lot of things in the bloodstream. Some of these things can be detected, some of these things can be mutated, some of these things the immune system might think they are foreigner things, and they're going to develop autoantibodies. So by having this theory, this hypothesis, from the findings that you see, you can select findings that they are mechanistic.
Great. Thank you very much, Leo. So second question is, in general, are serum sample better than plasma for Olink? So I can answer to that. Actually, if you look in our validation data, we are, let's say, we can say that plasma samples are preferable, but there is not a sample that is better than another. There is a preparation, a sample prep that's better than others. So it's very important to have all samples prepared in the same way, with very low pre-analytical variability. So like Leo presented, so it's very important how you prepare your sample. But it's true that plasma often is used instead of serum. But blood samples normally have the same quality if they are very well prepared.
I will agree with you, Pasquale. I also prefer plasma samples. Sometimes, I mean, the pre-analytical variability on plasma is releasing a very important biomarker that you cannot measure it because it's pre-analytical. It's affected by the pre-analytical variability. In these cases, you might have to switch to serum. So, but in general, yes, I think plasma samples overall have lower pre-analytical variability and are preferred.
Yeah, and I would also add, if you're using plasma, please be sure that you're using always the same anticoagulant in that. If it's ADTA, it has to be always ADTA. If it's heparin, it has always to be heparin. If it's citrate, it has to be citrate.
All those time now that we're using Olink, there is one advantage that is very sensitive and can pick different proteins. And this is an advantage, but when you start changing coagulant here, you can see like a very big effect. It's not because it's because of the technology that can pick biomarkers.
Exactly. So, I see popping up a lot of questions. Now we have 10 questions. I hope we can answer to all. Is there any relation with gut microbiota in disease variability and diagnostic accuracy? Very nice question. Microbiota is, I think, very, very important in all disease variabilities. I will answer on the Olink assay part. So we don't have bacterial biomarkers in our protein list. So everything we are measuring are human proteins. So we cannot do any microbiota study based on the Olink assay. We don't have the bacterial proteins in the library. But sure, you can integrate other tests.
Yeah, I mean, it is relevant. Yes, it's a great tool to have it in. But later on, I mean, depends on what kind of platform you're going to use. If it's going to be NGS based or if it's going to be protein based. I mean, if you can use it for biomarkers, yes, go ahead. But you have to think about the platform at the end of the day. And yeah.
Okay, next question. Do you see value in combining Olink's targeted protein panel with unbiased global protein to improve biomarker discovery, particularly when trying to identify novel mechanism or expand panel in emerging therapeutic area? So 100% agree. I mean, we are not putting us like in competition with unbiased global proteomics. You know, if you are using, for example, mass spectrometry, we are more like two pieces of the puzzle to be put together. So yeah, oh, sorry, that question away. Yeah, so you can combine both technologies, and actually the correlation are very, very good between Olink targeted approach, let's say, and classical proteomic unbiased approach.
I agree 100%. I mean, they're very complementary. Yes, especially for novel proteins and targeted mass spec is great.
Okay, next question is it's very technical. So Olink quantifies proteins abundances using PCR amplification signals. In short RNA sequencing, PCR-based, such as differences in amplification efficiency, etc., is corrected through normalization methods. Since Olink also relies on PCR, how does the platform address PCR-induced variability? Yeah, so very good question, technical question. So we are spiking in each well internal control. So we are normalizing actually the signal coming from each well for each sample and each protein. So, and we are normalizing specifically with the control that we spike for PCR, because it's where you can create the more difference. If you are having like an error in the pipetting, then you multiply because of the PCR. So yeah, we have a normalization method based on this control and on plate controls that we have on each plate. So we have a nice way to normalize the data, but we can go into details if you want. We can explain to you. So you will see in all publication with Olink these NPX values that are normalized protein expression values. And we can show you the calculation that we are performing. Next one, are you planning to layer any other omics data to this study? So Leo.
Yeah, definitely. I mean, we're doing transcriptomics. We would like to find the mechanism. So we're taking a piece of tissue. We would like to do some imaging as well to validate the mechanisms that we were thinking of. Not for advancing on a biomarker, but mostly to focus on the mechanism. For biomarker, we were thinking of using cell-free DNA, but there are some other issues that we need to think about the platform that we're going to use later on.
Okay, next question. Did you see any advantages using Olink Explore HT library in addition to the standard Explore platform? So we can easily highlight that the standard Explore platform was the one that we were using before releasing HT. So it was 3,072 proteins. So we have more than 2,000 protein difference between the two platforms. So Leo, you saw a big difference.
Yeah. We were lucky that you released the 5,000 later on, because when we started the project, the 5K was not, the HT was not present. So we have tried both. There is a big advantage, not only that they have more proteins, but also it's much easier to run. It's more robust, like as an assay, and it's much easier to run the 6K.
Yeah. Let's say we improved the workflow to be lighter and also ecologic. Let's say, using less reagents, but having the strong performances and I will also add that the proteins that we added in HT allowed us to cover full reactome pathways.
Yes. The other thing that we noticed, we were thinking with AI that we're gonna discover a lot of new proteins because we looked for anything that has been published. From what we understood, because if something is published, it means that there are antibodies, it's a well-known protein, that's why someone published something. So at the end of the day, with the HT, we didn't find a lot of new proteins. Most of the proteins that we found, it was already on the HT. So HT was covering a lot of the biological pathways that were interested. We found 58, that half of them was autoantibodies. Of course, you cannot find autoantibodies on the HT platform. So at the end of the day, we found only half of the 26, and that good proteins out of the 26 was only like 1 or 2. So at the end of the day, the HT covers very good the biological pathways.
Okay, next question is about the main advantages and limitation of Olink compared with high-resolution mass spectrometry approaches. Okay, so yeah, I think partially we answered already this question. So the Olink library, as you saw, it's an antibody-based method. So we are having a discovery approach, but the limitation is that you have the 5,416 proteins because we have the antibodies for. So all other proteins that are not in the list, you cannot see because there is no antibody. So yeah, this is something that you can still perform with mass spectrometry. The positioning of the technology are a bit different because since the beginning, Olink was focusing on the low abundant proteome. So we are very, very focusing on all these proteins that are very low abundant, and with normal spectrometry, mass spectrometry approaches, you have to deplete the high abundant and then do a lot of work on the sample preparation. When with Olink we can address low abundant, mid abundant, and high abundant at the same time. So again, for me, they are not competing each other; they are more complementary, and they can give you two picture of the biology that you can put together and correlate very well. Yeah, the next question I think is more on the presentation. Was the final predictive model that claimed 100% sensitivity cross-validated, or were there other efforts to avoid overfitting and ensure generalizable predictive power of previously unseen samples?
Yeah, very good question. I mean, it might be some overfitting. I mean, we presented the 100% sensitivity and the specificity just to show that we can capture the biology and we can identify biomarkers. Later on, it's not this sensitivity and specificity that we're going to go to the clinic. Later on, a lot of these proteins, we're not going to find good antibodies to develop. They might be cross-reactive. There might be a lot of reasons to drop some of those 50 different proteins. So at the end of the day, when that's going to go to a clinical use, it's going to be a lower number. But still, I mean, we start with a lot of proteins that can be used to create a final kit. So just wait like a few more 1-2 years to have the real sensitivity and specificity.
Yeah. And then the last questions, one is about the 28 protein hallmarks, if you can tell something about.
I mean, from these 28 proteins, I mean, you can make it for 50. I mean, there is because it's a ranking, it's a prioritization. So depending on how big is going to be your plex, how much money it's going to cost at the end of the day, and how many of these 28 or 50 are going to work, you're going to create a subset that at the end of the day will be used for the clinical use. These 20 proteins are different hallmarks of cancer. Some of them are very well known. They are overlapping with some, that was very nice, that they were overlapping some other published CRC data, and some of them are new. So,
The next one is more on the discovery pipeline, if you use machine learning, for example, to identify.
Yes. Sorry. Using the bioinformatics, we use bioinformatics machine learning in many different steps of the discovery. Not just to select, just to prioritize. one important thing is not to prioritize your biomarkers just by what has the best ROC, because you need a biomarker A and biomarker B not to be correlated. If all of them are correlated, they're not going to work together. You would like them to capture different kinds of disease. The ideal biomarkers is like a not correlated set of biomarkers that can capture the mechanism. And when one fails, algorithmically with machine learning, you can use the other set. And this is the best strategy.
Yeah. And last question. So we are in time, 2 minutes left. So after you identify circulating biomarkers, do you map those back into the cell, intrinsic signaling pathways, driving diseases? So
very important. We're still waiting for the transcriptomic data. We would like to correlate that with the transcriptomic data because these are biomarkers on the blood. We would like to see what happens on the tissue and select biomarkers on the blood that we see the mechanisms of the tissue. So we still need a few more months to answer this question.
Yeah, and I would like to add that Olink is also doing a big effort giving you the tools to analyze your data. So if you're running an Olink project, then if you have people in your lab working with R, we have an R package with all the scripts already prepared. And in one of these, there is the pathway enrichment script. So yeah, definitely.
We use it a lot. We use it a lot. Amazing.
Yeah. Exactly. Okay, so with that, I finished the Q&A list.
And great, thank you so much. Thank you, Pasquale. Thank you, Leonidas, and also thank you to Christos. Was really an engaging session, this one, and really great data, great case study, great engagement from the audience. I would like just to thank, our speakers and the audience to staying with us for this coffee break today. And I think that we can conclude the session. So, Sydney, over to you.
Perfect. Okay. Thank you all again for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Olink Proteomics, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will go ahead and alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, that is it for today. Thank you so much, everybody. Have a wonderful rest of your day. We'll see you again soon.